On behalf of CHI's Global Web Symposium and our sponsors for today's event, Rapid Novor and Absolute Antibody, welcome everyone. I'm Allison Proffitt, your moderator. Today we'll talk through various approaches to safeguard your research through recombinant antibodies, cell culturing procedures, antibody protein sequences, and reference identifiers. We have an exceptional panel today, and I want to get started, but to give you a quick overview, we'll start with Andrew Bradbury from Specifica, then Michael Fiebig from Absolute Antibody. Next up is Alicia Henn at Biospherics, then a joint presentation from Zach McDonald and Anthony Stagiar at Rapid Novor, and finally we have Anita Bandrowski of SciCrunch. Welcome to all of our speakers, and we'll start with Andrew Bradbury, founder and CSO of Specifica.
Hi. Today I'd like to talk to you about problems with the use of research antibodies and a way to overcome those problems using naive libraries, and in particular naive libraries which are really performing extremely well. So the problem I think you've all heard about already, but I'd just like to run through a few slides. So back in 2010, David Rim's group published a paper talking about antibody validation, and the highlighted section there, furthermore, for commercially available antibodies, it is clear that what is on the label does not necessarily correspond to what is in the tube, is an indication of the problems that we're all suffering from. Another from Dr. Saper, blind faith that the antibody will stain whatever the manufacturer claims is not consistent with good science. So the problems are that antibodies don't recognize the correct targets, and they also do recognize incorrect targets with off-target binding. So how bad can it be? Well, here's an example. Some of these antibodies, as indicated here and here, were sold as recognizing CDK1, but also recognized this additional protein called CEP152. Now, not all antibodies sold recognizing CDK1 also recognized CEP152. You can see some examples here on the right-hand side, but two of the most popular ones did. And in fact, those were used in over 200 published papers. Now, in general, it is reasonable to assume that many of these problems are associated with polyclonal antibodies. However, it's also true that monoclonal antibodies suffer similar problems. And so in a recent paper that we published a couple of years ago, we found, looking at a number of hybridomas, that a large proportion of them contained additional chains. So the majority, about 70% or so, did not contain additional chains, but about 30% contained additional productive light chains, and then there were 1 to 2% that contained additional productive light chains and heavy chains, or just heavy chains alone. Now, where there were additional chains, it was clear that the use of a recombinant antibody where the correct VH and VL genes were cloned out, expressed and used gave much better signals, as you can see for all of these examples here. So on the left-hand side in each panel you have the recombinant antibody, and on the right-hand side you have the original hybridoma antibody, and you can see that in each case the recombinant antibody is giving a cleaner signal with less background. However, surprisingly, and we don't really have an explanation for this at the moment, even when we found hybridomas that had single VH and VL genes, and we took those out, expressed them as recombinants, and now tested them, the signal at the same concentration of antibody was better in the case of the recombinant antibody in the hybridoma. So we don't know why that is, but it's something to bear in mind that even in the case of good monoclonal antibodies where there's only a single VH and VL, there may be additional problems. Obviously, if you have more than one VH or VL, then the problem is related to the fact that the incorrect VH-VL combination may bind off target and may therefore increase cross-reactivity. Now, so how much money is wasted? And so this was a commentary that was published with over 100 co-authors back in 2015. And at that point, the global spending on protein binding reagents was 1.6 billion, with about half of that in the US. So the annual spending on bad antibodies, assuming that 50% of antibodies are validated and researchers buy bad antibodies as often as they buy good ones, essentially about 700 million, approximately 800 million antibody money is wasted on spending of bad antibodies. And of that, approximately half is wasted in the US. Now, that was back in 2015. Since then, it's been estimated that the market has now increased. So now 3.5 billion. And so the annual spending is obviously even greater. So we're talking about billions of dollars here going down the drain on antibodies that don't work. And this takes no account of the time wasted by researchers, additional secondary antibodies, on the opportunity cost of having incorrect results in the literature and people trying to follow them up. So how do you solve this problem? Well, it depends who you ask. So the average customer will say we need better antibodies. The average manufacturer says nobody will buy anything other than traditional antibodies. And just going back to some of the great entrepreneurs of American history, so Henry Ford said if I'd asked people what they wanted, they would have said faster horses. And Steve Jobs said it's not the consumer's job to know what they want. People don't know what they want until you show it to them. And so this is part of the purpose of this presentation here. So that paper that we've published back in 2015 started a bit of a conversation on this topic. And we followed that up with a letter indicating that recombinant antibodies alone are not enough. They really need to be characterized and validated. They should also, if they're expressed recombinantly, in an ideal world, we would identify them by their unique barcodes, which would be their publicly available sequences. Characterization in that case would be required only once if everybody was using the same recombinant sequence reagents. What you'd need to do once you received a recombinant antibody with a known sequence is you just test the functionality, and then you can reasonably assume that all the other properties of the antibody, ability to work in immunofluorescence, immunoprecipitation, Western blotting, and so on, would be retained because we're talking about the intrinsic property of the antibody. Now, many companies did not like this response, and they also agreed that antibodies should be properly validated. We never said anything to the contrary. They took a different approach, which was identify good antibodies and reputable companies and just use those, which is a reasonable approach, but it's still an antibody sold by company A recognizing target X may have subtly different properties to an antibody recognizing target X sold by company B, and the results may not be directly comparable. So that's why we believe it's really important to be able to use antibodies that are defined at the molecular level by virtue of their sequences. Additional issues, companies said the journal should mandate disclosure of detailed validation data and antibody sources. Nothing to disagree with there. And they also say that public sequences would enable easy copying. That is a reasonable issue. I can't disagree with that. Perhaps there are imaginative ways of getting around that, where, for example, sequences are deposited in some sorts of databases which are only open to the companies, and clones are given unique attributes, unique numbers. So that even if people don't know the sequences, they're still able to know that they're using the same antibody. Now, this has actually gone a bit further. The European Union recently got involved. I was on an expert panel, and the final recommendation, this is a long document of over 80 pages or so. But anyway, the EU recommends that animals should no longer be used for the development and production of antibodies for research. And so EU countries should no longer authorize the development and production of antibodies through animal immunization. Now, the EU has a policy dating back to 2010 where if non-animal methods are as good as animal methods, then those non-animal methods should be used. And it was after that that the EU stopped allowing the testing of cosmetics on animals, and this is a practice which is now spread throughout the world. So furthermore, non-animal derived antibodies are well defined and better reagents that will improve the reproducibility and relevance of scientific procedures. Now this is something that we've been saying, but the original goal of this was based on the animal welfare aspect. Now the power of sequence-based antibody definition is that there's absolute reproducibility between different experiments. So I can repeat or reproduce your experiments using exactly the same reagents. We will never lose good antibodies. They will become immortal because the genes and antibodies can always be resynthesized. And as antibody citations accumulate, a complete understanding of the properties of any individual antibody will also accumulate. As I mentioned earlier, complete characterization is required only once. And in modern molecular biology, genes, mRNA, proteins, oligos, siRNA, all those things are defined by sequence. Why should antibodies be different when the technology exists to define them by sequence? Now, there are additional advantages to the use of sequence-defined binding reagents, not just antibodies. So one could imagine a situation where there's web-based distribution of antibody sequences through large machines that will synthesize genes and then do in vitro transcription translation. Such a machine has been published, although it's pretty large at the moment, but one can imagine things improving. You can put different FC domains from different species, and then you can also play around with the format of the antibody, adding additional functionality such as enzymes, fluorescence, and so on and so forth. Now, I'd just like to give a quick interlude to the music industry as a victim of disruption. And so starting back here in 1973, you can see that the most money was made from singles, and then most money was made from LPs, which is this green one, and then that slowly transitioned over to cassettes, then CDs. This is all the physical material. And then back in about 2005 or so, we started getting digital, excluding streaming in about 2010, 2012. We're getting streaming, and now streaming is the biggest source of music industry revenue. So there is nothing special about the antibody industry that it should not be and could not be disrupted. The music industry has been severely disrupted by the introduction of a number of different technologies over the years. So how do you generate easily generate effective recombinant? So everybody knows what good therapeutic antibodies should look like, as illustrated here. And good research antibodies are essentially very similar, except the issue of immunogenicity is not so important. And so what's more important is low polyreactivity. And a good selection platform that will provide antibodies with those properties would allow you to carry this out relatively rapidly and cheaply, and also have antibodies that are functional in research assays such as Western blots, immunohistochemistry, so on and so forth. So I have wanted to design high quality antibody libraries much of my career. Unfortunately, I was unable to do that within academia. I couldn't find funding for it. Unfortunately, things are different in the commercial world, and we've been able to. And so the Specifica Generation 3 library platform is based on the use of well-behaved clinical antibodies. Those antibodies coming out of the libraries are substantially liability-free. They have very high functional diversity, and we provide them in an exclusive fashion. So what does it mean when you select antibodies from these libraries? So you have superior therapeutics. Over 80% of tested antibodies have no biophysical liabilities. Over 60% of the tested antibodies have affinities less than 10 nanomole, and we're getting between 500 and 5,000 different clonotypes per target. So just going through how we made that library, or those libraries, very quickly. We started off with the best therapeutic scaffolds. We then identified and isolated natural CDRs by either next-gen sequencing or PCR. We eliminated those CDRs that had sequence liabilities, and then we embedded those liability-free CDRs within the well-behaved therapeutic scaffolds. So what you end up with is a library of antibodies where you can see there's a lot of different diversity all embedded within this identical same scaffold. So all these CDRs then are from real antibodies that are in real people. We use what we call replicated natural for all but the heavy chain CDR3. These are extracted from our data sets. We eliminate errors and sequence-based liabilities, and then the DNA encoding those CDRs is synthesized in arrays. Now, in the case of HCDR3, the diversity of the HCDR3 is too high to synthesize in arrays, and so what we do is we directly amplify from at least 1 billion purified B cells from at least 10 donors. And those will contain some liabilities, but I'll address those later. So the selection platform we use is we start off with our libraries in phage display. We have at least 10 billion different clones. And then we do a couple of rounds of phage. We transfer to yeast. We find this is extremely important to fine-tune affinities, specificity, and expression. And then we have two different approaches. In one approach, we just pick random clones, test them for binding, and then sequence them using Sanger sequence. But in an alternative approach, what we do is we do next-gen sequencing on the full sequence output. We do machine learning to identify clonotypes, and then we synthesize those genes. And there's some to and fro between these two methods. So in some cases, when you do next-gen sequencing and machine learning and you identify clonotypes, you'll find that representatives of those clonotypes are found in your random clone picking. So just to show roughly what a selection sort profile looks like. So this is after selection, a couple of rounds of selection in yeast. So the amount of display, which is the amount of antibody on the surface of the yeast, is shown in the x-axis. The amount of antigen bound by yeast displaying that particular antibody is shown in the y-axis. And so each one of these dots represents a yeast particle displaying the same antibody on its surface. And so what you're interested in is antibodies or yeast particles that are found in this upper right quadrant here. And you can see that when we stain the population at 100 nanomolar, 10 nanomolar, or 1 nanomolar, even at 1 nanomolar, we're getting over 40% of the antibodies in this particular selection are still binding the target. Compare that to negative control, where we're getting very little binding. So then when we take the outputs and we sequence them using next-gen sequencing, we find that we have very, very diverse clusters. As you can see here, depending upon the concentration that you use for the sorting, the number of clusters in this particular case ranges from 800 to about 3,500. Now, the way we define our clusters, each cluster is extremely different one to another. And so this is shown here for the HCDR3, where the average distance between different HCDR3 clusters is about 10 to 11 amino acids. And if you concatenate all the CDRs together, then the difference is between about 20 and 40 amino acids. So these are not just single amino acid substitutions, these are very different sequences. Now, if you remember right at the very beginning in this library, we eliminated all the sequence-based liabilities. And so if you look at what we call our Generation 1 library, which is one I made 20 years ago and which has served much of my academic career, you can see that less than 10% of antibodies being selected from that library have no sequence liabilities. And 3/4 of the antibodies coming out of that library basically have between 2 and 4 different liabilities. Compare that to antibodies coming out of the Generation 3 library, where about 3/4 of the antibodies have no liabilities, and basically the rest of them contain liabilities only in HCDR3, and of those liabilities, 80% only contain a single liability, sorry. So then when we look at the affinity of the antibodies pulled out from this library, this is directly from selection using phage plus yeast display, sorting down to 1 nanomolar, you can see that the affinities here are extremely good. So 650 picomolar, 1.9 nanomolar, 900 picomolar. You can see they're very, very good indeed. And straight from selections, here's 5 antibodies where we've done extensive analyses. You can see that the KD of just over 20% are sub-nanomolar, and the remaining 40% are between 1 and 10 nanomolar. So this shows that the antibodies are producing extremely high quality in terms of affinity. Now, what about developability? Because this was the other thing that we were keen to solve. And so the way we did this was we took the single chain, we turned it into a full-length IgG, we took representatives of each individual library, and then we tested them for a number of different properties, as indicated here, using a number of different assays. And then the test antibodies were the parental scaffold, and then 4 clones chosen from each library, as well as an antibody called sirukumab, which has known poor developability. And this is a list of all the antibodies in the clinic with antibodies showing what we call a red flag indicated as in a red color, and each column represents a different assay. Each row represents a different antibody. The parentals for our library are all up here at the top, and down here at the bottom we have sirukumab, which is our known negative control that has known poor developability. And so when we look at one example here, size exclusion chromatography of a clone from library A, what you can see is that first sirukumab behaves very strangely, has an aberrant peak. Also note that it has broader distribution representing an aggregate shoulder. There's more aggregation out also for sirukumab, and a little bit for both the clone and the parental. And then in general, the parental and the clone essentially, sorry, oh dear. I've lost that. The parental and the clone essentially superimpose one to another. And the difference between the parental and clone, just non-treated and frozen and thawed 10 times, show very, very little difference in size exclusion chromatography. So if we look at all the clones we pulled out from library A, then the values below, if they're light blue, that means that their mean is within the therapeutic range. If they're bright blue, it means that the mean is better than the parental range. And if they are red, it means that they're worse than the therapeutic range. So you can see sirukumab has a lot of problems. Abrilimab, which is the scaffold for library A, has, as expected, means within the therapeutic range. And then if we look at four clones taken from abrilimab, what you can see is that on the whole, they are either as good as or better than the parental abrilimab. When we look at all the antibodies we tested, over 80% of antibodies have no biophysical liabilities, as you can see here. And those that do have liabilities, they only have a single liability representing about 16%. So conclusions. The Generation 3 antibody library platform rapidly generates highly functional recombinant antibodies, very high affinity, very few biophysical liabilities. Now, this is very important for therapeutics. It's less important, I would argue, for research antibodies, but nevertheless, if you can make an antibody easily and you can concentrate it, that's an advantage, whatever the final use of it is. We get very high antibody diversity against different targets, and we use unsupervised machine learning algorithms to identify distinct sequence clonotypes. This is the Specifica team that was involved in the work. And if you're interested in learning more, then either send me an email, abradbury@specifica.bio, or visit us at specifica.bio. And with that, I'll say thank you and stop.
Thank you so much. Again, I want to remind all of our attendees, if you would like to send questions, please do so. There's a chat panel on the right side of your screen, and at the bottom of that, you can send to all panelists and type your questions there. Andrew, do you routinely obtain the affinities that you've described?
So where we've done extensive analyses, yes, this is what we see pretty routinely. I'll be honest, when we first got these affinities, I didn't believe them. We actually bought the Cartera plasmon resonance machine to use and measure affinities in high throughput. And I was convinced that there was something wrong with it. So we actually sent our samples off to Cartera to check them because the affinities seemed too good to be true. Usually, in my experience, scientifically, when results are too good to be true, they usually are. But in this case, it wasn't the case. So yeah, we're getting pretty good affinities as those shown.
Excellent. Can expression of recombinant antibodies exhibit differential post-translational modification, which would affect affinity between batches?
So it depends upon the antibody you start with. Certainly, it is possible that if you express antibodies that contain post-translational modification sites under different conditions, then those post-translational modifications can change the activity of the antibody. That said, the differences between the same identical genetically sequenced antibody expressed under different conditions, I would argue, the difference is going to be much less than when antibodies derived from two separate immunizations using two different targets are compared. So whilst there is possibility for some differences, I think the differences are going to be minor compared to classic animal immunization. Also, I'd like to add that you can now eliminate all post-translational modifications from your antibodies, particularly if you have the sequence and they're recombinant, so you can ensure that that's not a problem.
Okay. So what are the shortcomings of the SCF-VFC format over the classical IgG format?
So we find the shortcomings. There's one single chain FV format has one shortcoming, and that is that when you take the single chain FVs and you convert them to IgGs, not all antibodies will convert. Now, the conversion rate in our hands, depending upon the scaffold we use goes up to about 95%. So we don't find it's that much of a problem. We believe that the shortcomings, or that particular shortcoming, is balanced by the advantages of being able to display single-chain FVs both in phage and yeast. By being able to go from phage to yeast, you can then really play around with the properties of the antibody you want to select in terms of affinity, cross-reactivity, species, cross-reactivity, elimination of binding to closely related molecules, and so on.
Okay. Awesome. Andrew, thank you so much. We're going to move on. Michael Fiebig is VP of Product Portfolio and Innovation at Absolute Antibody. Welcome, Michael.
Hi, everyone, and thank you very much for tuning in to this webinar on reproducibility in research. My name is Michael Fiebig, and I'm Vice President for Product Portfolio and Innovation at Absolute Antibody. And I would like to tell you a little bit about how we think antibody engineering can help address reproducibility issues in research. To start off, let me give you a little bit of background about Absolute Antibody. We're an antibody supplier, CRO, founded in 2012 and have grown to around 60 staff. We were founded with the vision that everyone should have access to recombinant engineered antibodies, not just people working in biopharmaceutical companies, but also researchers in academic labs, as well as people developing diagnostic tests. The notion that all antibodies should be made recombinantly has really gained traction in recent years. And this is in no small part thanks to the article published by Andrew Bradbury and Andreas Plugtun in 2015. Absolute Antibody started off in Oxford in the UK, but we soon moved our production site to Redcar in the northeast of England. In 2018, we merged with the Boston-based Reagents company Carafast, where we now also have a logistics hub, as well as marketing and product licensing bases. In 2019, we moved our headquarters to Redcar and opened an office in Amsterdam in the Netherlands, where we focus on product development and customer service. In order to understand what the problem is we're trying to solve, we need to understand what are the kind of antibodies that are being used in research and diagnostics. And I've split this up into three different categories. We have polyclonal antibodies, we have hybridoma-derived monoclonals, and we have recombinant monoclonals. Polyclonal antibodies are obtained from the serum of immunized animals. They're great across many methods because they recognize different epitopes. The problem is that these antibodies are variable and they're finite. An animal will evolve its immune response the longer it is exposed to the antigen, and at some point there's no more serum you can get from an animal. And by and large, these antibodies are completely undefined. You really don't know what you have in them. Hybridoma-derived monoclonals, where you've taken a B cell that secretes an antibody and fused it with a myeloma cell, are another option. These are generally produced in vitro, and since you have one antibody, you recognize one epitope. The nice thing is you can grow these cell lines forever and ever and get as much antibody as you need. So you really have an infinite production. Now, as you might be able to guess, I've put some stars next to some of these points because that is sadly not always the case. Some antibodies some hybridomas will not grow in vitro. They need to be produced by ascites production, where you inject these into the peritoneal cavity of an animal. It's quite a gruesome process and, in my opinion, rightfully being outlawed in many countries. The problem with one antibody is that it's often not one antibody. Many hybridomas express multiple transcripts, multiple antibody chains, often making several different antibodies, and so they're not truly clonal. Stable, well, these cell lines are fusions with a cancer cell, with a myeloma. They're inherently genetically unstable, so you get genetic drift, which can lead to loss of productivity, but also changes in isotype and lots of other issues. And finally, infinite production possible. Well, antibody cell lines can be lost, they can die off, they can be contaminated. This is not a safe bet. When we're looking at recombinant monoclonals, we're really starting off from the sequence of an antibody. This sequence can be obtained by sequencing a hybridoma, or it can be obtained by direct B-cell sequencing, or the antibody can be discovered using in vitro methods, such as phage display, yeast display. Once you have the sequence, you clone it into a DNA vector, and you can transfect it into your production cell line. This can be a mammalian cell line, but also yeast, or it can be transformed into bacteria. What you get with these is a precisely defined product. Antibodies have an amino acid sequence that defines them. If you use the sequence, you will get this antibody. With this, you achieve the highest possible reproducibility, and it opens up further options. And it's these 2 points, these 2 last points, that I want to spend the time I have and tell you a little bit about what we can do with this. In order to make recombinant antibodies on an industrial scale, you need to master 3 technologies: antibody sequencing, antibody engineering, and recombinant expression of antibodies. When we first started off doing hybridoma sequencing, we were using a PCR-based method, and we were able to sequence about 300 hybridomas in 4 years. That's a lot of hybridomas, but there was also a lot of work, and this method was not scalable in order to be able to sequence the tens of thousands of hybridomas that are out there. In 2016, we switched to a total transcriptome shotgun sequencing approach. Think RNA sequencing in order to identify antibody coding transcripts. I've shown a flow diagram here on the right-hand side to illustrate the process a little bit. This allows us to sequence well over 1,000 hybridomas per year, and we probably haven't reached our full capacity on this yet. Not only can we do lots of hybridomas with that, but we can also sequence complex, non-clonal hybridomas and even rescue dead hybridomas. On the antibody engineering front, we currently make 15 different species of antibodies as IgGs, Ms, As, Es, and we can also make recombinant fragments such as Fabs, Fab2s. Moreover, we can take this further into making multi-specific antibodies and even go down the route of antibody humanization for biotherapeutics development. Recombinant expression, our workhorse, is a HEC293-based transient expression platform. We can also use CHO cells, but HEC293 is our go-to cell line. With this transient method, we can make milligrams to gram quantities in as little as 4 weeks. And that's quite useful for the reagent space, where you don't need kilograms, but you need lots of different versions of antibodies made at such moderate scales. The antibody engineering that I alluded to, I just want to illustrate this in this diagram. Once you have the DNA sequence, having been obtained from either hybridoma sequencing, B-cell sequencing, or in vitro selection methods, once you know the sequences of the variable domains, the antigen binding domains, you can either make a precisely defined copy of the original antibody, or you can change the constant part, the Fc regions of the antibodies, and essentially create anything that you want. So you can take a mouse antibody, turn it into a rat or rabbit, into a human, into an IgE, what have you. So once you have the sequence, the world is your oyster. Now, I already touched on a precisely defined copy of the original. What do I mean by that? I mean, we've identified the active sequences in a hybridoma and just made the same antibody again. Now, this actually shows a lot of improvement for many hybridomas because many hybridomas are not clonal. This is a study we helped author, and it showed that out of 187 hybridomas, roughly a third is not clonal. And so you're making an inactive byproduct. And this can be an additional light chain, can be an additional heavy chain, or can be both. And if you look at the data on the right-hand side, you can see that the recombinant material consistently outperforms the hybridoma-derived material. This is all published in this paper shown here by Bradbury et al. Now, the other thing to note about this is when you have multiple transcripts being expressed, as the cells undergo culture, there can also be fluctuations in the relative abundances of these transcripts, leading to a very variable antibody product. The next story I want to tell you about is how we rescued a classic reagent. We were working with this antibody that recognizes DNA strands that have been modified by the chemotherapeutic agent cisplatin. And very early on in its lifetime, this rat hybridoma was contaminated with a mouse hybridoma, sort of a tissue culture cross-contamination. Over time, this meant that less and less rat antibody was actually made from these cultures, and less and less active antibody was seen. Now, we tried to get original stocks that were not contaminated, but this happened so long ago that none of these uncontaminated stocks were available. We first tried addressing this by simply sequencing and picking lots of PCR clones. However, this did not work out. This was still before we used the next generation sequencing approach, and we really had a bit of a dead end there. We did then have an idea about purifying out the rat cells first, and we had to apply a few tricks there. In the end, we did manage to get there by a PCR method. At least we got a rat heavy chain. The light chain even looked a lot like a mouse chain, which is due to a specific rat allele. Anyway, it was pretty complicated, but in the end, we were able to get an antibody that we thought was the right reagent. And when we then functionally tested it, you can see on the left-hand side, this is the recombinant material, which shows a nice dose dependency in its response. Whilst the original material that people had been working with on the right-hand side is a bit more of a random response generator. So I hope you can see here that by going recombinant, we were able to rescue a very important reagent. So this sort of brings me to the wrap-up of the first little part of my talk, which I call the traditional reasons to go recombinant. And there, we're really looking at reproducibility on the level of consistency of the product that is made and being able to have continued access to such reagents. With this, we ensure that we have a defined and active heavy and light chain combination, and we can rescue contaminated hybridomas. And this can be contaminated with other hybridomas that have no bearing on the activity, or contaminated with microbes such as mycoplasma. This method also allows us to replace ascites production. And we've also seen that it can actually help you identify duplications in hybridoma collections. Just as much as you can cross-contaminate hybridomas, you can simply make a little mistake and put the same hybridoma in vials labeled for different clones. And since we license in a lot of hybridoma collections, we see duplications a lot, far more than people think that they would happen. And that then always puts into question, well, the validity of the research that is done with these two different clones? Do we really know that there are different clones? And sequencing also allows you to safeguard reagents for the future. And by that, also safeguarding the findings that were made with these reagents. And this is not just by random loss that they die off, but think if you're a researcher who's developed lots and lots of reagents during their career, what happens to the clones when you retire? More often than not, they just get thrown away. At the end of your career, someone empties out the liquid nitrogen and throws it all on the skip. And there are dozens of examples where really valuable hybridoma collections were just thrown away. And that's it. Moving beyond that, we can go recombinant to allow us to go further with our science. This is really looking at reproducibility in the context of an entire experimental system. So the options we have here are, for example, engineering an antibody to reduce unwanted or counterproductive interactions in an experimental system, or engineering an antibody to design complex controls with absolute precision that allows to validate an assay. Before this sounds too philosophical, let me show you some examples for this. The example to the first point I want to give is around immunologically clean reagents. Consider this. The majority of in vivo research in mammalian hosts is done in mice. However, the antibodies that are used there are generally of rat or hamster origin. These antibodies are immunogenic and will be rejected by the mouse immune system. This not only leads to adverse reactions such as anaphylactic responses, but also leads to short experimental timeframes because neutralizing antibodies prevent the injected antibodies from working, and also leads to inconsistent responses, something that is generally addressed by increasing cohort size and doing a lot of statistics. But maybe we can prevent this to begin with. What we did, we chimerized a rat. An example for this would be using chimerized rat or hamster antibodies to create mouse anti-mouse antibodies. The example I'm going to show in a second is a PD-1 blockade assay. So we've got a tumor cell, a tumor in a mouse, and the cells of this tumor are expressing the checkpoint ligand PDL1 and 2, and the T cells that are trying to mount an immune response against this tumor express PD-1. Now, the interaction of PDL1 and PD-1 leads to suppression of the immune response. There are antibodies, however, that can block this interaction and thereby lead to activation of the immune response, taking the brakes of the immune response, and to clearance of the tumor. As described, we applied a chimerization trick to this. So we took the original rat IgG2A antibody and turned it into a mouse IgG2A with an Fc silencing mutation. That means this antibody has no effector function. All it does is block the PD-1 interaction with its ligand. Now, this is a very classic experiment. Many of you will have seen experiments using the anti-mouse PD-1 antibody RMP114 in immuno-oncology papers, for example. And what the data generally looks like is like the red or green trace, where over the first few weeks you see very nice suppression of tumor outgrowth compared to the isotype control. However, after a while you see that even in those treated animals there is some tumor growth. More often than not, actually, the assay is terminated at around 30 days, so you don't have to explain what's going on here. In our case, as I said, we had the original rat version as well as an engineered mouse version, and we looked at different dosages in an assay where we're giving bi-weekly injections for well over 60 days as well. And what you can see is that the engineered antibodies that are shown here in the purple and blue trace show much better performance. Even between the different doses, the low dose of the mouse antibody performed better than the high dose of the rat antibody. But you see there are quite big error bars on there. And what I like to do is look actually what's happening on the level of individual animals, because large error bars generally mean a lot of variability or lack of reproducibility. And if we look at individual animals, you can see on the top we've got the animals treated with the rat version of the antibody. At the bottom we've got the ones treated with the mouse version. You can see that there's a big difference in the top left and top right. You can see some animals respond very quickly, others don't respond at first, then go back down, others go up very, very quickly, others slower. So you really need quite large cohorts in order to do this assay to get a representative readout. When you're looking at the bottom plots, you can see, well, yes, there is one animal in the 3 mg/kg dose that did eventually have a tumor grow. But apart from that, it's a very nicely reproducible and nice homogeneous response. This, however, is zoomed out quite a lot. Let's look at what's happening at these short time frames that people generally do research at. And even here you see, this is the same data, just zoomed in a little bit to visualize the first couple of weeks. And there you see at the top data, these responses are all over the shop. You really see a lot of variability, whilst in the bottom ones treated with the mouse version of the antibody, you get a very homogeneous response. And if you picked just a handful of mice out of this cohort, let's say you picked 3 or 4, you would actually get with the data shown at the bottom, you would actually get very representative data just on those. Whilst at the top, you really need to pick very large cohorts to get a meaningful readout. Now, doing this type of engineering is, of course, very familiar to anyone working in the biotherapeutics field. For human therapeutic antibodies, we also at least chimerize them, and now more often than not, humanize antibodies, just to prevent this kind of interaction in humans. But we can now also apply this to research reagents. Moving away from mouse assays to the slightly more topical human diagnostics, the calibrators and controls used in the development and in the running of diagnostic tests are often patient-derived material. Now, these can be hard to obtain, and especially considering infectious disease states, biohazard concerns do present themselves. And much like a recombinant antibody, these are finite and need to be pooled to reduce variability, etc. But they are also just very undefined. We have already said you can take any antibody and turn it into any isotype. So why don't we do that for this? For example, we can take an anti-coronavirus antibody and turn it into human IgM or IgG. In fact, you can go even further and actually define different IgG subtypes. So, for example, for COVID-19, human IgG1, IgG3 are very important, as well as IgM and IgA. And we can blend them at similar levels to be used as calibrators, or blend them at levels that might be representative of an infected individual, and so have a good serological control. And we actually did this. We ran some lateral flow rapid tests and added some engineered controls onto those, and could very, very nicely see both the good performance of the assay as well as the performance of our antibodies in this context. But this doesn't have to stop at infectious diseases. We can also think about allergies, where you might want to think about IgE or IgG4. And with this absolute definition, we can get absolute quantification of readouts. So in summary, recombinant antibody technology and engineering make research more reproducible. This can be on the level of a better performance in staining applications, or as illustrated when you rescue a reagent, safeguarding it for the future and allowing future generations of researchers to work with this reagent. But I think I've also shown you that engineering can help improve how an antibody is tolerated in vivo. And this not only maintains long-term performance, but also leads to far more homogeneous responses that are more reproducible between different subjects. And finally, how we can use antibody engineering to create precisely defined controls and calibrators for diagnostics, making sure that these assays are really meaningful and are running correctly. These last examples I showed you are all from our catalog. And what's maybe quite interesting is that we're not discovering new clones. We get new clones from collaborations or from licensing, and really taking in a lot of classic clones and giving them a new lease of life through antibody engineering. And as we grow this catalog, we're always maintaining this absolute approach about it, not just on the level of the protein that's made, but also in terms of the formulation, to make these very attractive reagents for the research community. But we are more than just a catalog. Our entire technology platform is available as services for those who can't do this in-house yet, particularly looking at large-scale sequencing projects, but also offering recombination of entire clone collections, which could be quite interesting for other reagent companies as well. We can also do humanization and bispecific and trispecific antibody engineering, which is very interesting for people in the biopharmaceutical space. But we're also applying some of these approaches to murine settings, like murine bispecifics, to bring back some of this antibody engineering to basic research. And you can see some of these bi and tri-specific antibodies that we make on this periodic table of antibody formats here. We've made all of these, so trust us to make a crazy molecule for you. Right, that's it from me. I would like to conclude by thanking the Absolute Antibody team, in particular Lisa and Morgan from our marketing team, for helping set this webinar series up and for helping me with making my slides. And thank you very much for your attention, and I'm happy to take any questions you might have.
Michael, thank you so much. We don't have a lot of time for questions, but so I'm not going to get to them all. But with all of this antibody engineering and switching of formats, do you find antibodies still bind in the same way when in a different format, or are there differences?
Yeah, that's a good question. We have actually tested a few antibodies in a lot of formats. For example, we turned the human therapeutic cetuximab into probably by now 50 different formats, and we've not seen anything that would really suggest a big difference in binding. If you change the valency, you can get some differences. But between IgGs, it's very, very consistent. You see very consistent performance. So changing FCs really hasn't given us any unusual surprises.
Great, thank you. Okay, we're going to move on to Alicia Henn, the Chief Scientific Officer at Biospherix. She will be our next speaker.
Thank you to the organizers for inviting me to speak with you today about reproducible conditions for cells and tissues. I'm from Biospherix, where we take the cytocentric approach to life sciences. And describing that, I'd like to start by doing something a little different. I'm going to ask you to close your eyes for a moment and mentally stand in the doorway of your laboratory. Now look around inside. What do you see? Do you see benches? Do you see chairs? Do you see tables? And I'm going to ask you what of the things that you see is truly designed for the needs of the cells and tissues, which are arguably the heart of everything that we're trying to do there. Now open your eyes, come back to me. A lot of people might have thought of this. They think of their room air cell culture incubator, where the cells spend a lot of their time. Now it's easy for people to get their hands in there, you can get your face right in there. But I'm going to argue that actually this is a terrible place for cells or tissues to be in a lab. First of all, there's too much oxygen in here for cells and tissues that are used to the physiologic oxygen conditions inside the body. Second of all, it's highly prone to contamination. There's that water pan in the bottom that's practically a swamp. and it's always, the cells face a constant contamination risk in here. And number 3, every time that you open that door to the incubator, whether it's you accessing your cells or whether it's someone else in the lab accessing theirs, all the conditions change inside. CO2 goes off, the temperature goes off, everything changes. So it's a highly variable environment for your cells. At Biospherics, we design our incubators so they open into a closed cell handling space. There's constant HEPA filtration of the atmosphere inside, and the gases come from compressed tank gases, which means we can compose that atmosphere to be physiologically relevant for the cells and tissues that we're culturing there. Now, in a traditional lab, when you take your cells from that room air incubator to handle them, you take them to a BSC like this. And again, this is a very people-centric space. It's set up well for people, the needs of people, but it's, from the cells or tissues point of view, it's hyperoxic, it's hypocapnic, and it's also cold. And of course, the cells face their highest risk of contamination when they're open in this kind of space. So again, this is a people-centric space. This is not a cytocentric space. Cytocentric cell handling space is closed. It's separated from all the variation and all the microbial risk that comes with exposure to room air. Now, in a cytocentric cell handling space, we have control of the gases, so we can have physiologic oxygen, we can have constant CO2, the atmosphere is at 37 degrees, the floor is at 37 degrees, so the cells are never out of optimal conditions. In fact, they don't know that they've even left the incubator. It's like you're handling them inside the incubator. Now, we all have to use equipment to handle cells, whether it's for cell analysis or cell sorting or some other sort of cell handling. But every time you take that equipment and put it in room air, you're introducing all the variability that comes with room air and all the risk to cells that comes with room air. So we've made our systems so that they can enclose any type of equipment that you need for handling or analyzing your cells, whether it's microscopes or centrifuges or bioprinters or even cell encapsulation or vial filling machines, you name it, we can put it inside a truly closed and safe environment for the cells. More broadly, the ex vivo system protects the whole process start to finish. So your cells come into the lab, they go into the ex vivo, they stay there, and everything you need to do with the cells is inside the safe space with constant conditions for the cells until you're done with them or until they need to leave the lab. And we've been working with ex vivo systems for over 8 years now. We have not had one single contamination event because they're just aseptic by design. They can be as small or as large as you need. You can put one process in the lab, or you can put the entire lab inside. It depends on what you need. We make modules that are custom to different pieces of equipment, like the huge TCAN pipetting robot you see on the left here, or the large bioreactor you see on the right here, which was designed for whole human lungs. And we make our systems modular so you can start in the research laboratory, start small, find the optimal conditions for your cells or tissues, lock those conditions down where it's cheap and easy to make changes, and then you can add on modules as your budget allows, as your cell or tissue process moves towards the clinic. You can bring in automation, you can add capacity, you can scale up, you can scale out until in the end you have a fully GMP compliant cell or tissue production factory, and you use the same modules that you started with in the beginning, and you have the same conditions that you started with in the beginning, which means that you avoid the tech transfer risks that comes with changing facilities as you have to scale up and scale out and move your process towards the clinic where it can really help people. So once you've locked down all the conditions that you need for your cells or tissues, you can then use those conditions every single time that you run your process, run your experiment for truly controlled conditions. And if you think about it, even in the pinnacle of technology, the cleanest clean room out there, you have the same sources of variation that come into your cell process, whether that you find in the most rudest, most crudest cell laboratory anywhere. You have these suboptimal transients every time the cells change environments. When you take them out of the incubator, the oxygen goes off, the CO2 goes off, the temperature goes off, the pH goes off. And in a clean room, because they keep those rooms cold, it really is even worse for the cells. And they keep them cold because of all the layers of clothing the people have to wear. This is truly a people-centric environment, not a cytocentric environment. The cells have shifted away from being the focus in these laboratories. In the cytocentric laboratory, you have full-time optimal conditions for the cells and the tissues, which are arguably at the heart of everything that we do. It's why we're there. There's no mixing of the room air with the cellular tissue environment, which also is a bonus for biosafety as well, in that anything inside also is not being communicated with the room air where the people have to be. Does it make a difference to cells? Well, I have a lot of data on this, and I'm not going to go into a lot of detail here, but just touch on some of this data. If you're interested, please contact me, and I can send you much more in the way details about it. But we've been working with bone marrow mesenchymal stromal cells for years now, and have shown that full-time controlled physiologic oxygen is far more beneficial for their growth than traditional cell culture room air conditions for them. You get more cell numbers, you get more cell divisions out of them before they senesce. And I have a lot of people that tell me, well, I've got this tri-gas incubator that I inherited from someone down the hall, and I'm just going to use that, and they'll be protected most of the time in that tri-gas incubator. I only take them out for a few minutes. So, from my point of view, they should be fine. But I'm here to tell you, and I can show you the data, that this is not actually fine. It's actually very stressful for the cells to be exposed to these changes in conditions transiently. MSC do not grow as well. Also, we've shown that there are fewer mitotic events after cells have been exposed to room air conditions, even for short periods of time, for a medium change or for cell passaging, whether that's early passage or late passage for bone marrow MSC. And once you put them back in that incubator and the numbers all come back to what they should be from your point of view, actually the cells are not at those conditions yet. The pericellular oxygen actually takes almost 2 hours to recover after a quick medium change in room air. And the intracellular oxygen takes even longer. It takes almost 3 hours to recover after a quick medium change in room air. So even transient conditions in cell or tissue environments can make long-lasting changes on the actual cell environment inside. Now, we all know that last year the Nobel Prize in Medicine went to the folks that discovered hypoxia-induced factor 1 alpha. And I just want to show you one thing from some of the early data characterizing HIF1 alpha. I just want to show you the scale on these figures. We're looking at single minutes for HIF1 alpha to be modulated in response to changes in cell environment, cell oxygen levels. And downstream of HIF are all these critical cell pathways and cell functions that can affect cells long term, even after brief changes in conditions. So once we get the ideal conditions for our cells or tissues and we lock those down, we can reproduce them every run for better reproducibility between experiments, but we can also take that same equipment, take those same conditions, and put them anywhere in the world. So now we have truly verifiably reproducible conditions between labs, between different places, whether you're in San Diego or Singapore or on a ship at sea, you can have verifiably the same conditions for your experiments. So when we gather to talk about reproducibility for life sciences and trying to improve it, which we all care deeply about, we talk about things like reagents, like the antibodies we've been talking about here today. We talk about things like record keeping, we talk about reporting issues, we talk about statistics, we talk about cell identity, and all of these factors come into play to different extents, whether it's you trying to reproduce experiments from your own same lab, whether it's a collaborator down the hall trying to reproduce your results, or whether it's someone halfway around the world trying to pick up a paper and reproduce your results. But the one factor that comes into every single experiment performed anywhere on planet Earth is cell conditions, which is why it's so important to bring this into every discussion that we have about reproducing biology. So we can take the capability of full-time protection for cells and full-time control of conditions, and we can apply it to reducing the variability from experiment to experiment, from day to day, from year to year, from user to user. We can take physiologic simulation, right oxygen levels that we find inside the body, and use it to reduce the variability in translating results from in vitro to animal or in vitro to human. And we can take that clonable modular equipment and apply it to reducing the variability from lab to lab, to location for distributed production, and from reducing tech transfer rates over time when we have to translate from the lab to the preclinical to the clinical. So I've got to stop here. I'm going to stop here. Please feel free to reach out to me and talk to me. I'd like to know more about what you're working on, and we can talk more about your specific situation. And please join us on the LinkedIn for the in vitro reproducibility group. Thank you.
Alicia, thank you so much. We've got some questions. Are the conditions inside the ex vivo system recorded?
Yes, absolutely. Those conditions are recorded 100% of the time. They're recorded in each module independently so that when you have your cell data, you have matching environmental data in detail that you can also publish with that.
Okay. Should journals require reporting of cell culture oxygen levels, do you think?
Ooh, that's a good question. With the capability of having all this detailed information about the cell environment, comes with that ability to record the oxygen levels and track them, I think comes the obligation then to report them and to make sure that when we compare studies between different labs that we're comparing apples to apples, because oxygen levels make such a tremendous difference in the findings that we get from in vitro assays. I think absolutely journals should require it.
So is there a best oxygen level for cells in culture? Is there a primary?
Yeah, that's a really good question. The best oxygen level really depends upon the cells and the tissues being studied, because there are so many different oxygen levels in the body that are appropriate to different tissues. So while all of them are lower than room air is never appropriate for study of anything inside the body. It's irrelevant to anything going on inside the body, because as soon as air is inhaled into the trachea, it's already mixing with air that's being exhaled. So the oxygen level drops quite rapidly. Even in the lungs, it's far lower than what it is in room air. So even if you're studying a lung air system, you'd be looking at oxygen levels closer to 10% oxygen than the 21% that you see in room air conditions. Now, if you're looking at something that's a tissue that's deeper in the body, like bone marrow, you'd be looking at far lower oxygen levels, somewhere between 2% and 5% oxygen. And if you're looking at something like chondrocytes, which are naturally avascular, they have a natural oxygen level that's almost zero. It's almost anoxic. So actually you want to go very, very low to have a physiologically relevant environment for that tissue type.
Okay. All right, so I've got some logistics questions. Can I put an ex vivo on an existing bench in my lab, or is this something that needs to be re-engineered?
Yeah, no, it's absolutely the case. We make these systems as big or as small as you need to fit into your existing lab. We can put them right on a bench top. They don't have to have the base that we make for them. And we try to be as flexible in fitting this to the space as we can. We've made ex vivo systems that wrap around a corner in a lab. So it's all made to fit your space, and it's all modular so that we can make it fit the cells and as well as your laboratory space.
Okay, great. How long does it take to get one? What's the turnaround?
Oh, it depends upon, of course, our queue. Our production queue tends to get longer at the end of the year when everyone's wanting to use up their grant money. But it only takes a few weeks generally to manufacture and test a system for you to send it out. So it could be anywhere from, say, 4 to 8 weeks to get your system, and it's very quick.
Okay, excellent. Alicia, thank you so much. We are going to move on. Our next talk has two speakers. Zach McDonald, Senior Scientist Manager of Proteomics Research, and Anthony Sejuhar, Director of International Business Development, both at Rapid Novor.
Hello, everyone. Welcome to this next talk in our mini-series about reproducibility issues in antibody research. I'm going to be your first host here, Anthony. I'm the Director of Business Development here at Rapid Novor, and in a minute you'll be hearing from my colleague Zach, who's our Senior Scientist and Manager of Proteomics Research. We've got a quick outline for you here. I won't get into too much detail here, but essentially we're going to give you, a couple of open-ended questions. Has your research already been affected by these kinds of issues? A little bit of an overview on, protein sequencing using mass spec. Some of the more technical details, and then getting into some use cases and what it takes to actually get a project started. So if you're in this talk with us today, there's a good chance that reproducibility issues have already affected your research, right? And I'm sure it's no surprise to hear that significant sums of money have been wasted on antibody research in the past, and probably even within your own research projects as well. And one of the cases that we've seen, within our client base is that we had a lot of wasted research in early stage kind of therapeutic development and basic research. So the examples that we've got here that we have a scientist from the NCI at the NIH, and she was doing some early stage kind of preclinical research on a new therapeutic candidate, and they had spent quite a bit of time and effort in quantifying some of those results using an antibody. And unfortunately, after a couple of years of actually conducting those preclinical assays, they'd run out of that initial batch that they purchased that antibody. And when they went back to the vendor to source more of that antibody, they found that the new batch of the antibody was actually different, right? And so whenever they conducted any of those preclinical evaluations, again, they found different results. And so effectively, all of the results that they generated thus far were no longer reproducible. And we have similar cases that we've seen as well, like the following, which was from Dr. Prasas and Dr. Diamanis, who are actually pretty close to us locally. They're from the University of Toronto, Mount Sinai group. And what they had done is they were actually trying to use an ELISA kit that they had purchased to quantify a particular protein of interest. So this was the CUB and zona pellucida-like domains protein 1, this CUSD1 protein, and the ELISA kit was sold to them in good faith, and they believed that they were using an ELISA kit to quantify that protein. But at the end of the day, which this day was long, it took 2 years, and it cost about $500,000 of their own dollars, they found that this ELISA kit was actually quantifying a different protein. It was quantifying the antigen CA125. And so a lot of the research that they generated up until that point was obviously difficult to finish because they couldn't get consistent and reproducible results. And I think that ultimately, this is what they found was that those results were, I guess, permanently irreproducible because they were in fact quantifying the wrong protein. And the entire point of their initial investigation was look at this CUSD1 protein when in fact they were studying the wrong protein using the ELISA kit. So this was a note to beware, and that these kinds of reproducibility issues are certainly common throughout the industry. And in another study here, which has been pointed out in the past by some of our co-speakers here, we see that Lukanovicus et al. Conducted a study in which they were investigating the protein activity of some centrosomal proteins specifically the CDK1, as well as the CEP152, CEP63, and CEP57 proteins. And what they did is they created a SNAP tag fusion protein. SNAP is like a small little protein that can attach a fluorescent marker. They attach that marker to this fusion protein in order to quantify the readouts. And so what you see on the top channel here is basically that SNAP is there, and we can see it because we measured it. More importantly, we see this part here, right? So this is what we're looking at. And so this bottom half here is actually showing us the activity of this anti-CD1K channel against, well, what's in the tube. And in this case, we can see that the Santa Cruz antibody actually performed well, and the Apcam antibody performed well at binding against not only CDK1 but also the CEP152. And whereas the antibodies on the right-hand side here, the Sigma, the Apcam, and the BD antibodies all bound specifically to the CD1K SNAP fusion protein. And so what this is really showing us is that it took them 5 antibody purchases to get the 3 that are actually binding specifically. Whereas the first 2 are actually much more popular in the literature. They're much more well cited. A lot more discussion surrounding them, despite the fact that they're not binding specifically against their target. And in fact, if you're using it to quantify CDK1 in this kind of fashion, you're going to have a tricky time actually reproducing those results. And that was specifically why they wrote this paper. Interesting note as well is that that Sigma antibody is actually a poly. I just kind of found that interesting that these first 2, actually the rest of them are all mouse monoclonals, whereas the Sigma is a rabbit poly, and it's still binding specifically against the target, unlike the other 2 on the left. Okay, so let's say you even eventually, after doing this kind of research, find, okay, great, now we've got a specific antibody. Well, unfortunately, that's not enough, because even if you have a specific antibody, like I pointed out earlier with the NIH researcher, you could still have issues in terms of sourcing that antibody going forward, right? Because it could be sold out. And if you've actually inherited this project from someone else, you could also have similar issues. Because let's say you've acquired a catalog of antibodies from a collaborator, either internally or from an academic institution. You could have issues in terms of actually growing those cells because they've maybe been sitting in a freezer for 20 years, and try culturing that, or you could not have it cannot be easy to actually sequence the genes out as well for similar reasons. They've just been sitting in a freezer for 20 years, and now you're actually trying to do some sequencing work on that, and that might be beyond your capacity as well. So this is where we really feel that we fit in here, because what if we could just sequence the protein directly? Well, this is where we fit in, right? So Rapid Novor has, over the past few years, become a world leader in protein sequencing using mass spectrometry. We've been in the software, proteomic software industry for almost 20 years. Has been in the game for a while. And so himself and Migi and Shishian, our co-founders, really created Rapid Novor with the intention to sequence monoclonal antibody proteins as well as proteins more generally, and in a high throughput fashion. That's always kind of been one of the bottlenecks here is, how accurate are your results? Are they actually accurate? And, can you do this in a reasonable amount of time? And I'm happy to say that, up to this point, we've got over 300 clients worldwide, including institutions like MIT, Harvard, University of Cambridge, the real Cambridge, as well as, UCSF and many more internationally, as well as many of the major pharmaceutical companies. They use us because ultimately they have mass spec teams, but, I think that it makes a lot of, and it makes a good economic calculation to work with us and to just because of our throughput. And this is kind of where we've directed the company from its inception. Is always try to think of it as we can sequence any protein of any species anywhere. I think more commonly though we've targeted our analysis against monoclonal antibodies, and that takes the shape of IgA, nanobodies, fragments, like whatever it is, we can sequence it. Fundamentally, if it's made of amino acids, we can sequence it. It's always just a matter of how much time and money is this going to take. But as you can see in the case of monoclonal antibodies, we've done a really good job of consistently sequencing them in a robust fashion. And a quick little run through on how that looks. We start off with a sample preparation using 5 different enzymes, and then we take that those digests, which will chop up your antibody into smaller peptides. We fly those peptides on the LC-MS, and once they've flown on the mass spec, they hit the analyzer, and the analyzer will then read out a mass over charge ratio. We can convert those spectra into peptide sequences, and once we have many different peptide sequences, we can then assemble them into a full-length sequence representing the antibody or protein of interest. And I'll let Zach jump in next and get into some of those more technical details.
So thanks, Anthony. In the next few slides, I'm going to give a light technical background to what we do and highlight the strengths of our sequencing platform. So our main work is centered on the use of high-end, high-resolution mass spec. These machines allow us to get extremely accurate masses, or mass of charge, of the fragments of the proteins we analyze. The raw output of the MS is a spectral graph, displaying M of Z on the x-axis and abundance on the y. The fragment masses allow us to build up the amino acid sequence in a process known as de novo sequencing. If you take a look at the slide in the top two spectra, we see a textbook case of de novo sequencing. The spectra is simple, and by calculating the change in mass between the peaks, we can determine the amino acid sequence. Great job. The sequence accounts for all the peaks, and you are a star sequencer. Of course, the real world is always more complex than this, as can be seen in the real spectra below on the slide. The raw data is noisy and can have many unmatched peaks, even with the best sequence. To deal with this complexity, smart algorithms and informatics tools are essential when sequencing proteins in a high throughput manner. De novo protein sequencing by mass spec is definitely not something new. It's been done since the 80s. What is relatively newer is the informatics tools that we use to assist us. Without these tools, manual de novo sequencing is just too slow. So there are a number of tools available to the community. These include algorithms such as CHAMPS and Supernova. But in the special case of antibody sequencing, a challenge for all these tools is obtaining 100% coverage and 100% accuracy. Of course, if you get one amino acid wrong, that can adversely affect your binding. And yes, I'm quite biased, but we do believe that we have one of the best informatics tools and platforms for protein sequencing in the world. It is based on the novel algorithm developed by one of our founders, Ben Ma. It incorporates modern machine learning and AI. It allows us to process our data, analyze our data, and produce results very quickly. The platform also allows for low confidence determinations to be easily flagged. These are then picked up by our bioinformatics team, and they are investigated and resolved. So using this platform, we are able to produce high confidence data. With the foundation of strong informatics, we also benefit from a very active wet lab component in our company. This has allowed us to adapt to and overcome many of the challenges associated with protein sequencing. In the special case of antibody protein sequencing, we need to deal with post-translational modifications. These modifications change the mass of the amino acid, so it's important that we are aware of them. We also need to analyze and process the data quite quickly, so the informatics tools and the easy access to the wet lab allows us to do that quickly. Our samples are also not standard, so we have to deal with contaminants such as BSA, which we do quite well. Antibodies can also originate from non-modeled organisms such as camel, shark, llama, and this requires some custom wet lab sample preparation. There's also the challenge of certain amino acids which are isobaric, namely leucine and isoleucine. These amino acids are not distinguishable purely based on mass, and they require deeper level of iron fragmentation to reveal the presence of diagnostic ions. And this we do in a process known as wild. It's always fun to describe a process called wild. I'd like to say that I came up with the name, but no, it was somebody much smarter than me. So wild, it stands for W-ion, isoleucine, and leucine determination. So after sequencing a large number of antibodies, we can confirm that most antibodies contain leucine and isoleucine residue in their CDRs, and more than 50% of them contain multiple residues in their CDRs. So obtaining confident determination of leucine and isoleucine is an important part of accurate antibody sequencing. Without going into too much technical detail of how we determine leucine and isoleucine, well, first we know that if you look at leucine and isoleucine, and very good mass spec, it's still the same mass. But they are slightly different. And using the right instrumentation with the right method, we can release certain ions that distinguish leucine from isoleucine. And these are the W ions. Applying the right instrumentation in combination with peptide modification technology that we have developed in-house, we can reliably release these W ions and confidently assign leucine and isoleucine. In the next few slides, I want to show some data demonstrating the performance of our platform. Naturally, if we convince you to send us your antibody, which may be your precious last bit of antibody, I'm sure you'd want some comfort knowing that we do get the sequence right. Hopefully, this data will show that we do. So we looked at a comparison between our de novo protein sequencing and DNA sequencing. This was done on a relatively large scale. We looked at 20 chains from 17 myeloma cell lines. The sequencing, the DNA and protein sequencing, was done independently before we did the comparison. For each of the 20 protein samples, we did 5 LC-MS runs. This produced thousands of MS spectra, which included ATD spectra, ETD spectra, and ETHCD spectra. These are different kinds of cleavage energies which give different spectral information. We obtained high quality data, the mass error below 3 ppm for most spectra, and we were able to sequence all the expressed heavy and light chains with high confidence on the REMAB platform. The figure in the slide shows the sequencing coverage for one of the cell lines, OCI MY5. You can see this shows the depth of coverage that we obtained from the de novo sequencing. Of the 20 cell lines, we do not have DNA sequencing data for 3 of them, but of the remaining 17 cell lines, we were able to compare the DNA reads to protein sequences by DNA mapping. The DNA reads were translated into amino acid sequences before alignment. If we look at the figure, I know this figure is not easy to see, but we have the protein sequencing result right at the top, and below that we have the DNA reads translated into amino acid sequences. What this shows is that for each amino acid, we had confirmation with tens to hundreds of DNA reads. So this gave us strong confidence in our protein sequencing result. In this slide, we show an example of when the platform was used to detect an error in published sequence. This is the published sequence of the Waters intact antibody standard. So this data visualization is what we get on our informatics platform. It allows us to go into low-level detail of the data. And we can see the published sequence on the left of the screen, and the sequence, the ambiguous sequence was the SIT. When we processed the data that we have of our mass spec, we found that the evidence for the SIT sequence was pretty low. There was not a lot of high confidence spectra supporting the sequence. Using our platform, using our methods and analysis, we were able to get a new sequence which matched up much more closely with the spectral data. And this was the replacement of SIT with TIS. So we were able to correct this error. Following on from the previous slide, this figure shows the raw spectra obtained for the sequence. In the bulk spectra, when we apply the published sequence, we can see that there are a number of unmatched peaks. So this is unmatched information. When we apply our corrected sequence to this raw spectra, we can see that those unmatched peaks are accounted for, and this gives us strong confidence in this corrected sequence. That is the last slide from my side. Can you see all the amino acid matches? No, that's not the intention of this slide. What we want to show here is the confidence that we have in our protein sequencing. This is a visualization of our data. Each line represents a de novo sequenced protein fragment or peptide. These peptides are assembled, and we are able to deduce the protein sequence. The large number of these lines indicate that for each of the amino acids that we determine, we have a large amount of supporting information. So we can have a high confidence in each of those determinations. And that will give you a sequence that you can trust.
Thanks for that, Zach. So from here on out, we just want to run through a couple more use cases and then kind of show you guys the logistical workflow behind actually sending us a sample. So like I said, we're going to be running through a couple use cases. Specifically, we're going to look at therapeutic antibody development and rescuing existing antibodies that you might already have. Okay, so a common use case that we've seen is actually, once you've got your binder, how do I work with it some more? Because like we pointed out earlier, specificity of these antibodies is a really tricky parameter to ensure for. So once you've ensured that the antibody you're working with is specifically binding against your target, hey, how can I work this into other kinds of situations? And one that we found quite commonly actually is in bispecific antibody development. So if you have the ability to ask your vendor or your producer for permission, they might grant that permission with respect to using some of their antibodies for research purposes only. And what that looks like is in a bispecific antibody development program, because there are literally hundreds, if not thousands, of different ways you can make bispecific antibodies. You might want to just test if these binders, these known binders, can work in your format and target your, and work well in targeting your end molecule, whatever that is. And so we've seen this where, someone comes to us and they ask us to sequence the antibody, then they end up taking it with permission internally developing their bispecific antibody platform to conduct some kind of proof of concept study, like, does this antibody format work in this application, yes or no? And it's easier for them to do it this way, because instead of doing their own, antibody development campaign and spending a lot of time and money to do that, you can just work with off-the-shelf compounds, again, with permission, and then, test their antibodies in their proprietary bispecific antibody development platform and see if it works. And in other ways, let's say you have a known binder against a target and you just want to see if it if it works in different pathways or not. So you might switch it from an IgG4 to an IgG1 or whatever the format might be that you're interested in working with. It's definitely something that we can do, and luckily we've got the partners to do it. A little shout out here to Absolute Antibody. I say that the guys are doing the engineering work, not us. And here's another one. So certainly, let's say you've actually inherited that catalog. Like I alluded to this earlier, but let's say you're an academic researcher. Someone in the faculty is retiring, and they've been working on antibodies for years, and they're done. They're getting out of the game, and they want to hand off those antibody projects to someone else because they don't care anymore. They might hand it off to you, and at that point, you can try to work with that antibody because you might find that, hey, this actually binds really well against my target, but I can't sequence it because the genetic material is just not good enough because it's been sitting in a freezer for 20 years, and I can't culture these cells because, well, for some similar reasons, right? So in that case, again, looking at the antibody molecule directly might be able to open up that path forward for you. And so what does it take to actually get a project going with us? Well, we just need 100 micrograms of the antibody. That's really it. We just ask that you ship that 100 micrograms of the purified monoclonal antibody protein. Again, it needs to be monoclonal. We can't work with monoclonal and friends here. It needs to really be a monoclonal antibody. You ship that to us in our lab outside of Toronto, and we can return that report back to you in 2 weeks flat. And if you have many experiments you're looking to run with us, just let us know, and we can always talk to you about timelines. But we can usually up to 10 in as little as 3 weeks. And so really the key takeaways that we wanted to walk away from here were that reproducibility is a difficult issue, right? Like it's not trivial to ensure reproducibility of any of your results when it comes to using antibodies in them. So acknowledging that, it's important to safeguard your research against kinds of issues before you start investing a lot of time and money into them. But maybe something slips through the cracks, as you're doing your research, and you spend a few years in the trenches, and then at the end of the day you can't source the antibody that you've been working with for several years. Well, we're still here. So that's all we're trying to say here is that in these kinds of cases where the money's already been spent and research time's already been allocated, you need to really ensure that you can come back and reproduce those results. You can safeguard your research using de novo protein sequencing here at RapidNovo. So thanks for your time. Like I said, my name's Anthony, and Zach spoke earlier. And from here on out, we're just looking to take a few minutes to do some Q&A. So we welcome your questions.
Thank you both so much. We will start with a question from Michael, our previous speaker. He says, not sure if I missed it, but how many different digests do you tend to have to do to get full coverage? Or is this variable with projects? If so, do you know what factors influence this?
Yes. So routinely we do 5 different digests, and we try to keep this quite constant. This allows us to give the overlapping peptides that we need and allows us to do the assembly that we need. But yeah, so we don't find that it's necessarily variable. There are cases where we can sequence well from 1 or 2 of those digests, but routinely we will do 5 digests of different enzymes of our sample.
Great. Okay. Can your team sequence conjugated or labeled antibodies?
Yes. So occasionally we do get called on to do this. What we found is that with the labeling process, it's usually not complete. So there's usually enough unmodified peptides for us to work with. So yes, the answer is mostly we can sequence this type of antibody.
Great. How does your sequencing service work for polyclonal antibodies?
Yeah, I'll take that one. So as we pointed out in this talk here, this is primarily our remab for monoclonal antibody protein sequencing. And so this is that solution. But indeed, we have been working on for years a polyclonal antibody sequencing solution. And in fact, some promising results have just recently come from that. So if you do have a project that is polyclonal, it is quite possible that we can assist in that, but it would be a different solution in order to address that. Yeah.
Okay. So how do you ensure 100% accuracy for your sequencing protocol?
Yeah, good question. So essentially, as we've shown in the one slide where we, claim that we have the most, most sequencing reads per microgram of antibody, we hinge our accuracy on overwhelming abundance of observed data and in those peptides that we actually pull off of the mass spec. So in every single project that we report to a client, we have a minimum coverage of 3 to 5 peptides per amino acid, including the CDRs. But more commonly, especially if the sample is actually pure, like the NIST sample that we showed earlier, we can get dozens, if not hundreds, of overlapping peptides over every amino acid. But again, it really comes down to the purity of that sample and how much we actually have to work with.
Okay. So can RapidNovor deal with other antibody formats other than IgG?
Yeah, good question. And the answer is yes. We've worked with many different antibody formats, IgG, IgA, IgM, different variable fragments as well. So indeed, yes, we can work with other antibody formats. And in fact, other proteins which are not antibodies are also fair game for our analysis.
Right. Zach and Anthony, thank you so much. I also wanted to, before we move on to our last speaker, it does look like we'll have a little bit of time. So if any of our attendees have questions that they did not get added or we didn't get to, it looks like we might have some time for that at the end. So I just want to remind you that you can send your questions to all panelists on the chat bar on the right of your screen at the bottom. So closing out our speaker panel today will be Anita Bandrowski, founder and CEO of Cycrent. Welcome, Anita.
Hello. I wanted to talk to you today about why you should support RADs. The big question in this webinar series is centered around reproducible science. And my portion of that is just to kind of bring this down to a place where we can all really understand it. So if you're looking at any reproducible science, you can see that scientific paper as like this, like a recipe, where the first thing that you want to look at when you find a recipe is, well, what is the stuff that I need to gather? And then the second step is, how do I then put all of the ingredients together so that I can make a delicious dish? So if we look at another one of these recipes, we find that in this particular MBio recipe, we have an antibody, which is listed here as mouse anti-flag from Sigma. And so this is how a lot of people do talk about a particular antibody. They tell you what kind of dilution. But if I am a person who wants to recreate this recipe, I want to go to Sigma, and I actually find 17 different antibody products. And so, unfortunately, without an identifier, this is not a reproducible recipe. So the question is, can this really be solved? And in 2012 and until 2014, a whole series of workshops were held at the Society for Neuroscience meeting and at the National Institutes of Health, where we got 25 journals who would finally ask their authors to provide RIDs for this 3-month project. And this was supposed to be a pilot project. Only 2 of those journals started on time, but in fact, we are currently in the 5th year of this particular pilot project. It turns out that no one actually stopped doing it. So what do these things look like? And I think a lot of you have now seen, hopefully, what an RID looks like, but here's a nice example. So this is actually taken from the methods section of a particular paper, which is, excuse me. This is a method section of this paper, and this is what's called a star methods section. And here you can see that there's this particular antibody that's being described by this catalog number and the RID, and those are of course hot linked back to the antibody registry. The top journals in and around various fields, including kind of general biology, but mostly in neuroscience, do actually provide or ask the authors to provide RIDs, and they actually provide links to all of those RIDs. This provides a lot of interesting information about how those products are used. Lots and lots of journals in general do this, so this is one of those kind of generic STAR methods tables. These are available in all Cell Press publications. eLife, you should have seen a lot of these papers also with RRIDs. Journal of Neuroscience, some of our other society publishers do push this. And so overall, this is getting to be a much larger list, but unfortunately not as many journals as we would like actually enforce RRIDs. So if you want to try this at home, you can do this with any RRID, figure out where it's being used by which of your colleagues. This particular antibody is one of the, it's the new-one antibody. It's a fairly common one from Sigma millipore. And so, right now, according to Google Scholar, there are 559 papers that have used this particular antibody. So if we want to look, and we can kind of generate an idea or get an idea of how many people are actually using these RRIDs in their papers, we can estimate that in 2020, it's about 20% of the citations for this particular antibody that are actually using this reagent and using the RRID syntax for this reagent. There are lots of companies. This is Thermo Fisher, and there are many other antibody companies. You may have seen some of the mouse companies that have actually, so this is a project out of UC Davis. The MMRC is also telling authors how to use the RRID, and in fact, they have a nice little button to copy it. But is it making a difference across the literature? So here we looked at 316,000 different references to antibodies out of about 2 million papers in the open access literature. And we plotted the number of those that essentially had the catalog numbers versus those that didn't. So in 1998, here you have about 10% that actually where authors had used a catalog number. And then that is a very steady and slow increase till you get to about 2015 or 16. And then this overall line actually increases quite a bit. It's not quite at 50%, but nearly half of the authors are using catalog numbers. And this uptick in the usage of findable antibodies, the ability for us to be able to actually click on something and figure out what that antibody is that we're using, is being really driven out of the Cell, the eLife. These are all RRID enforcing journals. So these really are driving this big increase in antibody identifiability. And by the way, anyone can do this today. Just go to rrid.site or type in RRID into your favorite browser and you will find it. Okay, so why don't all journals do this? I mean, this seems like a no-brainer, right? Why shouldn't all journals enforce RRIDs? And in fact, the top journals do. We just got Nature and Science on board within the last couple of years. A lot of the society journals are on board. But unfortunately, this process of going after authors, asking them for yet another thing, it takes some time on the part of the journal. So, it costs them, they estimate, about 30 minutes per paper. And some journals don't have that amount of time. So these high throughput journals like PLOS ONE or tiny journals, they just don't go after authors at the same rate, even though, RIDs are part of their instructions to authors. So we thought we could maybe solve this problem by using some really cool text mining tools. And so we created SciScore. SciScore is a tool which takes as input the methods section of a paper, and ideally this would be used before the paper is published. It detects sentences that actually have antibodies in them or describe antibodies. It tries to suggest RIDs to authors if it actually detects a catalog number. And it checks whether those RIDs that are provided are correct. So Further, it does the heavy lifting for editors and for reviewers trying to check whether those antibodies are findable or not. And we're very happy that One of the big society publishers is now using this tool in all 9 of its journals, so this should drive an increase in antibody reproducibility at the AACR, which is the American Association for Cancer Research. This is what that new journal submission looks like, so this is brand new, just within the last month or so. Any new paper that comes into one of the AACR journals, there's an extra question which asks to put the method section into a part of the form, and then there's an email notice, and the authors are presented with the SciScore report, which contains this key resources table. So in this case, this particular author had actually used an RRID, and this is a relatively rare problem, but sometimes people make a typo in the number, and in this case, this number did not resolve, so this says unresolved. This is the thing. But you can see that the author had used actually Santa Cruz Biotechnology, and there's the catalog number. So based on that catalog number, we're able to actually suggest the right one. And so this is the, this is the 95, and it's the author had put in that it was 96. So, it can help address these kind of little typos and things that happen as part of maybe creating a large, long list of antibodies or other tools. When an author uses a cell line that's either problematic or misidentified or contaminated, the same kind of warning also goes up. So this is not the case only for antibodies, but the tool actually recognizes a lot of different kinds of issues. And again, it does warn authors when there's a problem that's detected. So where can we go from here? Well, it turns out that SciScore is, and a lot of really good product data that comes from good quality antibody companies, can actually be used together. So when an antibody company says that one of their antibodies, let's say antibody X, is actually validated in a particular, for a particular method, so in this case Western blot, and let's say it's validated for use in mice, but in fact a paper comes in where the same antibody, and you can imagine this is an RRID antibody because that can then go look up this information. Now the tool can say, hey, the authors are using this same antibody for immunohistochemistry and they're using it in CARP, right? And so what the tool can do is the tool can say, hey, authors, this is probably off-label use. You might want to be aware of that because this is not validated in the same system and for the same technique. So we need to work with all of the journals to identify all of the reagents. We're not going to stop with, and rest on our laurels just because we got Science and Nature. But in fact, we want to make sure that every single author in every single journal actually uses antibodies in a way that's very consistent, and at least you can find what those reagents are. So this is my shout out, my call to all of you. Can you help us improve science? So the way that you do is you can add RRIDs to your next paper. You can ask for RRIDs when reviewing a paper. And you can always go to sciscore.com to see if it could lend you a hand in being able to do that. Okay, thank you.
SciScore, is this something that's, I guess, how laborious is it for them to incorporate in their review process?
So most journals, thank you for that question. So most journals can do this today if they wish to. They can do it manually, but we're actually working with the backend providers, 2 of the major ones now, which take care of the submission and the review process for those journals. And essentially what that does is it makes the process really seamless. There's no, other than a new report that comes back, kind of like a new review, it's pretty seamless for the authors and the journals. That way they don't really have to do anything.
Okay. What can antibody suppliers or manufacturers do to make RRIDs more widely used?
So thank you so much for that question. That's great. We have so what we can do, to kind of support this initiative, is in fact to go in and pull the RIDs that are there, put them up on the website. So you saw the Thermo Fisher one. I think there are a lot of companies which are trying to do this and kind of are in process, but there certainly are multiple companies that already have that. BD, I think that's still in process, but a ton of them, like Synaptic Systems has it in the material data sheet. BioLegend has it on all of their product pages. So, essentially, please talk to me. I'll be happy to provide you with the RIDs that are already probably part of your products if you don't have a good list. If you do have a great list, let's work together to make sure that this data is as accurate as possible. I mean, we have probably 20 authors coming to us every day to register new products, and that's somewhat because we don't have all of the most accurate data from all companies. We do have it from many of our partners, which is great.
Great. And so in a sort of community supporting project like this, who is bearing the cost of SciScore?
So SciScore is actually a commercial product that charges journals to use that product. As part of the submission process, journals will charge authors a certain amount of money. Tiny fraction of that goes back to SciScore. And but there is a free version for authors to use, so that's a pretty straightforward one. As long as the author logs in with their ORCID, then they can get a small-ish number of runs every year. So that it's intended to, and actually the cost was borne by the grants that we had gotten from the National Institutes of Health.
Excellent, thank you. So we have a few more questions that have come in, and again, I encourage all of our attendees to keep sending those in. If you do, maybe tell me which speaker you want me to direct a question to now that this is an open floor. But we have a few minutes left, so I want to get to some of these. We're going to start with Michael. If you're ready, Michael.
Yes.
In the in vivo data, great. In the in vivo data, one mouse treated with the engineered antibodies still showed tumor growth in the end. If these antibodies aren't immunogenic, how can this happen?
Oh yeah, that's a very good question. So there are two possible explanations for this. One is that maybe simply this mouse developed a PD1 treatment resistant tumor, and this happens that tumors do actually evolve under the pressure of treatment, and so maybe here we're actually seeing something that could be scientifically quite interesting to investigate. The other explanation, and I personally actually think that's the more likely one, is that the mouse actually started developing antibodies against the antigen binding sites. And we have to remember that these in chimeric antibodies, the antigen binding site is still of the original species. So in this scenario, it was actually still of rat origin. And even though rat and mouse antigen binding sites look a lot alike, it can still be immunogenic. So then you started getting these maybe even anti-idiotypic antibodies, and that could really account for this growth. And yeah, these things can happen.
Thank you. Okay, I've got a question for Anthony. What is the fragmentation technique for generating W ions from isoleucine and leucine?
Sure. In short, it's called ETHCD, but I'll let Zach continue that a bit more.
Sure, yeah. So with the generation of these ions, you need quite a soft fragmentation process. So it's a fragmentation process called ETD, electron transfer dissociation, and that releases Z ions, and then we are then from that able to release the W ions. So it's a combination of ETD and HCD. We also needed to do some peptide modification to ensure that we can see those W ions on a regular basis.
Great, thank you. All right, my next question is back to Michael. The diagnostics industry has a bit of a reputation as being slow to adopt new technologies. What are their responses to the kind of engineering of serological controls you propose?
Well, so we've been trying to pitch this approach to diagnostic companies for years now, and I have to say, we were met with a lot of skepticism for a very long time. The current coronavirus pandemic has really changed a lot in this regard because things needed to happen fast, and we're working on this global effort to allow us all to be able to go outside again. And we've really seen changes in attitudes to engineered controls if they allow us to make more robust assays and allow us to do this a lot faster. And so it's a changing landscape, and it's perhaps one of the only positive things I've seen come from this, that people are more willing to look at new solutions. And yeah, that's what we're seeing, a lot more acceptance of novel engineering approaches.
Well, that's certainly good news, especially in this situation, this environment. We need all the flexibility we can get.
Absolutely.
I believe that is it for us. Thank you all of our speakers, so much for a very enjoyable session, a lot of information. I want to thank all of our participants as well for your questions and being a part of this. We have recorded the session, and we will be sending this out. So if anybody didn't get all their notes down, you'll have a chance to look at this again. Again, I want to thank everyone on behalf of PHI's Global Web Symposium. Have a nice day.